Hi, guys. Today we started by reviewing what we did yesterday. So we're going to spend a little time reviewing that, and then we're going to talk about today's lesson. So please watch very carefully and watch this whole video before you start the homework, okay? All right, so yesterday we talked about how cells are really, really, really tiny and small. We can't see them. They're the smallest living thing that exists. They're building blocks for our bodies and for living things. They come in different shapes and sizes. They're shaped like what they do, so they're shaped like their function. And living things can be alive and have one cell, which is called unicellular, or multiple cells, which is called multicellular. All right, let's look at these pictures right here. And I'm going to make it a little bit bigger. Okay, if we look at this, you'll notice that here we have two cells. There are two basic types of cells. So if you're a cell, you're either the one on the left or you're the one on the right. And there's a name for each of these. But what do we notice right away about these cells? Well, first I notice the shape is different on the left versus the right. The one on the right seems a little bit bigger. The left has a tail. The one on the left seems really simple on the inside, where the one on the right has a lot of different stuff on the inside, including this big circular thing on the inside. So basically, if you're a cell, you're either a prokaryotic cell or a eukaryotic cell. Let's look at the prokaryotic cell on the left. Prokaryotic cells are things like bacteria. They're single-celled organisms, so they are much less complex. If you notice, they have DNA, which basically is the building blocks for us. It's our code for what our body and our different body processes do. DNA is kind of all this green squiggly stuff in the center. So if you notice, DNA is just kind of floating around in the prokaryotic cell. A eukaryotic cell, if you notice, is a little bit larger. It's not the same shape. It has a more kind of a circular shape. It's got this big thing called a nucleus inside of it. So this giant structure in here is called the nucleus. And then it's got all these little things around it. DNA in this cell is located inside the nucleus. So as we can tell, the eukaryotic cell is a lot more complex than the prokaryotic cell. DNA is in this special warehouse called the nucleus. DNA in the prokaryotic cell is just kind of hanging out and floating around in the simple one. We are eukaryotic cells. So I always remember you are a eukaryotic cell. And that should help you remember that you and I are this kind of cell. We're more complex. We have a nucleus with DNA in it. So here's what we're going to do today. We're going to go to Google Classroom. And on Google Classroom, you should see for today. So if you go down to Tuesday, 11/1, you should see the link that's orange. Okay? On this orange link, once it goes there, oops, and mine showed mine. So hold on, let me go back. Once you click on the orange link, you should see this. It should say student. You're going to first enter the teacher username, which is KPICICCO 219. If it doesn't work, it means you probably spelled it incorrectly. So double check that it's KPICICCO 219. Then you're going to click on it. From there, it'll have you pick your period and your class, and it'll have you put in your password. Your password should be your first two initials of your name. So like mine would be KP and then a number. This is what you wrote in your assignment notebook. So if you can't find it, email me and I can tell you what it is, okay? But that's what your password is your first and last initial and then two numbers after it, okay? Eventually, let me go to educator here. Let me sign in really quick. Okay. Eventually, you're going to get to a screen. Come on, Gizmo. Sorry, give it a second here. You're going to get to a screen that looks like this. And you're going to click on cell types, and then you're going to launch it. And you're going to get to a lady that's sitting next to a tree. Okay, this is what you should get once you get there. So again, if you're having trouble, let me know, but this is what you should get to. This is what your gizmo should look like. Okay? All right. Let's talk about what your gizmo actually should look like on paper. So I'm going to go to this, and whoops, you see me, but I want you to see this instead. So now you have to look at me. Sorry about that. Okay, so now what you're going to do is what I want you to do is you can cross out this first part and this first part. Woo! Okay, go to the next page. All right. So basically what we're going to do in this is we're going to look at different cells, okay? So if you look over here, we're going to look at mouse skin and fly muscle, so all the way down the list. And you're going to write down 3 parts, they're called organelles, 3 parts that you see in the cell, and then you're going to tell me whether it's eukaryotic or prokaryotic. If you remember, eukaryotic means there's a nucleus. So I would write that down to help myself remember. Has nucleus. Prokaryotic, there's no nucleus label. So here's what we're going to do. Let's go down to a lotia leaf and I'll show you an example. And you can fill this in as I'm doing it. So the first thing we're going to do is we're going to go back to our Gizmo and we're going to click on Show All Samples. So we click Show All Samples and I'm going to go to a lotia leaf. It's a plant that's in the water. So if I click on it, you'll see a lotia leaf shows up. After that, once that's showing, I'm going to click on microscope. And now we're going to be able to look at it under a microscope. So the way that this works is that we have to focus it exactly like when we focus a microscope. So I have to start with the lowest power first and use the coarse focus. There we go. And I got it really, some of these are tricky, so I have to really make sure it's focused good. Okay. Then I go to my fine focus. Let me make sure this one's a little better first. Okay. Okay. Then I'll go to the 100 once I've done the coarse and the fine. And under the fine, under the 100, I'm only going to use the fine focus. So I want to make sure that's really good. Okay. Then when that's focused, I can go to 400 and just use the fine focus. Okay. Then I'm going to click on Show Labels, and I should be able to see labels. What you're going to do is you're going to write down 3 parts. And I know you don't know what those are, and that's okay. But if one of them is a nucleus, I want you to write that down. So here's what we're going to do. I notice nucleus. I notice cell wall. I notice cell membrane. So I'm going to go back to my chart. And under a lotia leaf, I'm going to write those 3 things down. I saw a nucleus. I saw a cell wall, and you can write this down too. And I saw a cell membrane. Okay, so I wrote down 3 things. Okay, now if it has a nucleus, it's a eukaryotic cell. If it has no nucleus, it's a prokaryotic cell. So if I look back at what I was just doing, I notice that there is a nucleus right here. So it must be a eukaryotic cell. So I'm going to put a giant E here. If there were no nucleus labeled, then it would be P. It would be a prokaryotic cell. Okay? So what you're going to do for all of these is go through and tell me 3 parts you see after you've focused it and gone to the highest power. Then you're going to tell me whether it's an E or a P. Then down here, you're going to answer this question. And then you're going to do and answer this part. And then you are all done. That's your homework. Please don't hesitate to let me know if you have any questions. Have a great one.